Good evening, everyone. We'll just wait for a few more minutes to let the others join and then we'll start today's session. Hello all, I think we can start our session today. Let me just share my screen. Is my screen visible to you people? Okay, I think it is. Okay, thank you. Good evening everyone, and welcome back to week 4 of the NPTEL online course Drug Delivery Principles and Engineering, the PMRF TA sessions. And we'll start with some of the key points that were discussed in week 4 lectures in the course. So to begin with, we have been discussing about a bunch of drug delivery systems, and we started this journey with discussing about what is polymer drug conjugate. We were talking about this thing, right? So let's say you have a very hydrophobic drug and you want to deliver it to the patient's body, but being hydrophobic, there are chances that the drug might precipitate before and it won't be soluble in the bloodstream, where it has to be soluble in the patient's body to reach its active site, the site where it's supposed to be is required for its action. So in that case, what you can do, you can conjugate this drug, this small molecule, with certain polymers which are hydrophilic in nature. And let's say, for example, polyethylene glycol, PEG. And then once you conjugate your drug with this polymer, it will make the drug hydrophilic because of the PEG chain surrounding this drug. And then you can deliver it to the patient's body, which will then be easily soluble. So that was one example of drug delivery system, which is the polymer drug conjugate. Next, we discussed about reservoir systems. So what are reservoir systems? These are a very saturated, a very super saturated, high concentration drug core over which we have a polymer membrane. And this membrane, so the main point about reservoir system is this membrane is non-erodible. So the way the drug will get out of this entire system is through diffusion. So you have a very high amount of drug in the core, and over which you have a polymer membrane which is porous, and over time water will flow. Once you deliver this system to a patient's body, water will flow into this, since it's surrounded by a lot of aqueous environment. Because of diffusion, because the concentration outside this device is very low compared to what is there inside, so the drug will naturally flow from higher concentration to lower concentration because of laws of diffusion. So that was one other example of drug delivery system, the reservoir systems. And then we saw metric systems. So what are metric systems? These are similar to reservoir systems, but instead of having the drug only concentrated inside the core, the drug is very well distributed all over the polymer. So these matrix systems can be erodible, can be made out of erodible polymers or non-erodible polymers. Give me a minute. Right. So so erodible as well as non-erodible metric systems. So the main advantage of having an erodible metric system is because you'll have to do surgery only once when you place that device or this system, delivery system, into the patient's body. Because now it is erodible, it will eventually be broken down into its constituents, and that would then be eliminated out of the body. So you don't have to do another surgery to remove this particular delivery system, which you have to do in a non-erodible matrix system. So once the drug has been released, its activity has been done, the treatment duration is over, you have to remove this non-erodible matrix system out of the patient's body. So that was another example of a drug delivery system. And then we saw extensively studied extensively about hydrogels. So hydrogels are nothing but these are swelling kind of polymers, right? So you have your polymers which are either connected with physical or chemical bonds, but in a dry state they have very small volume. They the moment they come in contact with water, because they have hydrophilic linkages, the bonds which are there in the polymer, the groups which are there in the polymer, they attract water. In this process they start swelling. So what you can do is you can encapsulate your drug inside these hydrogels and dry them up, store them. Once you want to use it, you can inject these hydrogels in the patient's body, and then they'll swell. The pore size of these polymers of these delivery systems will increase, and the drug will then flow out of these systems. That was another example. And the point to note here is that all of these systems that we have discussed until now are macro systems, right? They require surgery to implant into the patient's body, and in some cases they require another surgery to remove as well. So as I was saying, all of these are macro systems. So next we are going to discuss about micro systems. So these are nothing but your micro and nanoparticles. You must have heard a lot about this term of nanoparticles being used in drug delivery, nanoparticles being used for cancer therapy. Nanoparticles are also used for diagnostics to know whether a person has cancer or not. So you must have heard about this term a lot. So what are these particles? So these are nothing but, so microparticles are drug delivery particles which are in the micron range. That is from your 1 micron or more than 1 to 100 micron. On the other hand, you have nanoparticles which are nothing but particles which are in the nanometer scale. So these could be between 10 nanometer to 1000 nanometer. So depending upon your application, whether you need microparticles or nanoparticles, you can synthesize them accordingly. So the main advantage of these particles is that they're injectable. You don't have to do a surgery to put these particles inside the patient's body. Since these are of a very, very small size, they are easily injectable, and that's how they're used. And as I was saying, the main advantage is you can avoid surgery, and they also decrease non-specific delivery. Yeah, so various different kinds of drugs can be delivered via micro or nanoparticles. These could be your small molecules, meaning your antibiotics are small molecules. These could be your proteins. They could also be peptides or nucleic acids. And the way these particles are delivered is you encapsulate your drug molecule in these polymers, and they're of either micro or nano size. Or what you can do is you can make this polymer particles, and on the top of this you can then attach your drug. So there are two ways to deliver drug via micro and nanoparticles. One, you can either encapsulate it or you can adsorb it on the particle surface. Then you have to extensively characterize these particles, because since these are of a very small size, there are chances that they might elicit an immune response. So you need to know, you need to do a thorough characterization of these particles. You need to know the particle size and their shape. All of this then has an effect on how the drug is getting delivered from these particles. There is a concept known as polydispersity. We have seen polydispersity of polymers, right? So it's the same concept. Polydispersity of microparticles or nanoparticles tells you what is the size distribution of these particles. So whenever you have a formulation, it's not 100% uniform. Not all particles in your formulation are going to be of the same size. There is going to be some amount of variation that happens because of the way these particles are being synthesized. So there will be some degree of polydispersity. All we can do is we can try minimizing this variation in the size. Then we need to know the carrier composition, what polymer it is made up of, what is your drug, how much amount of polymer you are using in these micro nanoparticles. Also, there is another concept known as encapsulation efficiency, which means that how much amount of drug is actually getting encapsulated inside these particles. And then we have loading levels. Loading levels tells you in a particular micro or nanoparticle what is the percentage of the drug actually in this particle. So we'll talk about these concepts based on some of the questions in today's assignment as well. I'm just going through the points, some key points that were discussed in week 4 lectures, which will help us in solving the questions ahead. Then a lot of discussion has been done on particle synthesis in these lectures. Particle synthesis could be done with a single emulsion method. It could be done with double emulsion method. We'll talk in detail about these methods as well. And there is something known as spray drying. You use a very specific instrument known as a spray dryer to have these particles. So what you do in a spray dryer is you mix your particles, you mix your drug with polymer, and you spray it through a very tiny nozzle at a very high pressure. So what happens is when the liquid comes out of this particular nozzle, it will already make particles, and then there's a very huge drying chamber where your particles will then get dried. So as I was saying, you have your drug that gets encapsulated into this polymer, and when it goes through this drying chamber, the solvent will evaporate and you will eventually get dried particles. That is one method to have micro or nanoparticles. So spray drying will usually give you a little bigger microparticles. Then you have another method such as gelation, hot melt, etc. You can read about the details of these methods in the lectures itself. Then at the end of week 4 lecture, we saw something known as liposomes. This is another drug delivery technique. So what are liposomes? These are made up of lipid bilayers which encapsulate drugs. So you must be knowing that all the cells in our body, they have this lipid bilayer, right? So liposomes are synthetically made lipid bilayers which have drug inside their core, and because their lipid bilayer is of the same constitution that our cells have, the bilayer that our cells have, they are usually more biocompatible. Right, so all of these drug delivery systems that we have been discussing, they are foreign materials to the patient's body, right? So there are chances that they might elicit immune response. So in that sense, liposomes are very advantageous because their lipid bilayer has the same constitution as that of our cells. So our cells don't necessarily consider the liposomes as foreign. That's why they have high biocompatibility. The only disadvantage with liposomes is it is very difficult to scale up these. For a lab scale study, for research scale, liposomes are okay, but if you want to manufacture liposomes as a drug delivery technique for a number of patients, then it becomes a little bit of a problem. And also, since scale up is not very good, it consequently leads to higher cost. So these are some of the disadvantages of liposomes. So with these particular points, I think we can start looking at some of the questions for week 4 assignment. I'll display the questions on the screen. I'll read through the question and give you people some time to go through the options so that you can try answering yourself. If nobody responds, then we'll discuss the answers in a while. So let's start with the questions. The first question is, which of the following particles will be cleared the fastest by kidney? And we have to choose 2 correct options. So read through the question again, read through the options. I'll wait for a few minutes and let me know if anybody knows the answer. You can either to answer, you can either unmute your mic and let me know the answer, or you can also try writing it in the chat box and I'll read through your messages. Can everybody see the question displayed on the screen? I hope it's visible. Okay, looks like we have the first response. Somebody saying option 3 and 6, which is cationic and 1, 2, 3, 4, pegylated. Okay, thanks for answering. Does anybody have any other response other than option 3 and 6? We'll discuss about the answers in a while. Thank you for responding. Would others like to answer? The question is, which of the following particles will be cleared the fastest by kidneys, and we have to choose 2 correct options from positively charged, negatively charged, cationic, anionic, neutral, or pegylated. Okay, no other response. Let's look at the answer then. So the correct answer is positively charged and cationic. So whoever answered one of your options was correct. The second correct option was positively charged. So let's look at this answer in a bit more detail. So this is how the architecture, let me just start the pointer. Yeah, this is how the architecture of kidney cells are. So you first have an endothelial glycocalyx layer, then you have this GBM layer, and then you have this podocyte glycocalyx layer. So all of these 3 layers in the kidney cells are heavily negatively charged, right? So if you have a particle which is already negatively charged, this layer being more in negative charges, it is going to try repelling your negatively charged particles. So what would happen is, let's say if you have a weakly negative particle, there are chances that it might pass through the kidneys. If it carries a very high negative charge, it is going to be repelled by this particular layer. If it's a neutral particle, again there are more chances that it would pass the kidney, the cell layer. On the other hand, if you have a positively charged particle, more number of these particles would be attracted by this negatively charged layer, and there are very high chances that these ones will get cleared from the kidneys at a faster rate. So what was the question? The question was, which of the following particles will be cleared the fastest by kidneys? So if you have a positively charged particle, you have your first answer right at the first option, and also cationic, which means nothing but particles which have a positive charge. So both these particles would get cleared by the kidneys at the fastest rate. Pegylated will not be, because pegylated meaning that you have peg chains all over your particle. So these peg chains try increasing the size of these particles. So that is one of the reasons why people do pegylation over particles. Pegylation is nothing but addition of polyethylene glycol to the membranes, to the drug surface, and because now it increases the size of these particles, it is difficult for the kidneys to clear them. So that's why the correct option is positively charged and cationic. Is this clear? Let me know if the answer is unclear, or if there are doubts with the explanation, and we can try going over the question once again. At the end of the assignment, I'll anyways go through all the questions again. So you can also let me know the doubts at the end of the assignment. So are there any doubts with this question, or shall we move on? Okay, somebody had already answered 1 and 3. Thank you for answering. Any doubts with this question? I guess not. Okay, let's look at the second question then. The next is, what do you understand by the term encapsulation efficiency? Is it the weight percentage of the drug in the particle formulation, or it refers to the rate of dissolution or erosion of the particles under storage conditions? Is it the percentage of the starting encapsulant, that is a drug, that is captured in the finished polymeric particles? Is it the percentage of polymer, drug, solvent, surfactants, and any other additives remaining in the final particle formulation? Or a measure of how broad or narrow the size distribution is? Is it the amount of polymer per gram of drug encapsulated, or the polydispersity of the particles that are suspended in liquid? So what exactly is encapsulation efficiency? You have to choose one correct option. I'll wait for a few minutes and let you people answer first. Any volunteers? Somebody is answering option 3. Option 3 is the percentage of the starting encapsulant that is captured in the finished polymeric particles. Okay, thank you for answering. Anybody else has another response? We were just talking about encapsulation efficiency in the intro slide as well. So does anybody remember? Some people are trying to answer in the chat box. Okay, someone else is also saying option 3. Thank you for responding. Anybody else has an answer other than option 3? I guess not. Okay, let's look at the answer. Yeah, both of you who have answered in the chat box are correct. The correct answer for encapsulation efficiency, it is the percentage of the starting encapsulant, that is a drug, that is captured in the finished polymeric particles. So what does this mean? Let's say you started, you have 100 mg of the drug, 100 milligram, and you were able to load only 60 milligram of this drug in your polymeric particle. So your encapsulation efficiency would be 60%, because only 60% of the original amount of drug could you could put into the polymer. So why does encapsulation efficiency matter a lot? Is because now you, instead of, so you have to do some dose calculation before giving it to the patient, right? So instead of now having 100 mg of the drug, you only have 60 mg of the drug. The rest of the material is the polymer material. So accordingly you have to adjust the dose, because now you'll have to give a little bit more of the drug, because now you don't have 100 mg of the drug, you only have 60 mg of the drug. So that's why it's important to know what is the encapsulation efficiency of your particles. So here are some of the other definitions that matter a lot when we are studying about these micro and nanoparticles. So as I was saying, you need to know the particle size and shape. So there are ways to calculate it. First, you can have a qualitative assessment of these particles. This is done through microscopic techniques, but since these you can do it using light microscopy as well. That works for microparticles, but your nanoparticles are of a very very small size. So in that case you have to go for electron microscopy techniques to visualize the particles in a better way, or you can do a quantitative assessment in which you try to see what is the exact size of these particles. And also this can be done using light scattering techniques. They also give you the size distribution, which is the next point, the polydispersity. It is calculated from the size distribution. It tells you how dispersed, or how, what is the size of first of all, what is the size distribution of your particles. Are most of the particles of the same size, or is there a lot of variation in the sizes of your particle? Then you have carrier composition. So this just deals with the percentage of polymer. So whenever you have micro and nanoparticles, it is not just your drug, right? Your drug is encapsulated in a polymer. So you need to know how much amount of this formulation is your polymer, how much amount of this formulation is your drug. There might be some of the solvents you are using along with this. There could be some of the surfactants you use in the formulation. So what do surfactants do? They help you in avoiding aggregation of these particles, or there could be any other additives in the final particle formulation. So all of that comes under the category of carrier composition. Then the question that we are dealing with, encapsulation efficiency. It is nothing but the percentage of the starting drug that is captured in the finished polymer particles. You have another term to characterize these particles, which is the loading level or encapsulation ratio. So this is the weight percentage of the drug in the particle formulation. Let's say the weight of your particles is 100 mg, and amongst this the drug itself is 60 mg. So your loading level would be 60 per 100. That is 60% of loading level. And then you also need to check whether these micro and nanoparticles, are they stable or not. So stability can be termed in described in two different ways: physical and chemical stability. So chemical stability refers to the stability of the drug inside the particle over time, right? So what it means is, it's not like you are going to make these micro and nanoparticles and immediately use it on a patient, right? You don't know when the demand would come for your drug delivery system. So you make these particles and you keep it in storage for a while, until and unless it is asked for. So in that storage, your drug should always be active. It's not like you have stored your particles for 1 year and after 1 year your drug will stop working. Your drug has to be active. So that is what is meant by chemical stability. On the other hand, what is physical stability? So as I said, in these micro and nanoparticles, your drug is encapsulated into a polymer membrane. So your when it is sitting in the storage, when these particles are stored, it shouldn't happen that your drug is already leaking out of these systems. In that case, your particles will have a poor physical stability. So you need to know these things to make a very good formulation. They shouldn't the particles shouldn't start eroding while they are in the storage state. The drug also has to be active. So all of these things matter when we are trying to deliver these particles to a patient's body. So coming back to the question, encapsulation efficiency is nothing but the percentage of the starting encapsulant that is captured in the finished polymeric particles. Is this point clear? Or are there any doubts with respect to this question or the other few concepts that we saw? If any doubts, then I'll wait for a minute and we can discuss. Is this clear? Okay, then I'll move on towards the next question. The next is, in single emulsion solvent evaporation method of particle synthesis, what is the use of surfactants? We have to choose one correct option. So why do we use surfactants? Surfactants, is it to increase surface tension? Is it to reduce surface tension? To increase mechanical energy or decrease it? Or to help make particles of bigger size? So what is the use of surfactants in single emulsion method? If anybody knows the answer, please let me know. You can type your answer in the chat box, or you can unmute yourself and let me know. Anybody wants to answer? Someone is saying option 2, that is to reduce surface tension. Anybody else has another response? Thank you for answering. Okay, no more responses. Yeah, you're perfectly correct. The correct answer is to reduce surface tension. So what happens in a particle in single emulsion particle synthesis method is, you have your aqueous phase. So this is where you have water and surfactant, and in the other phase, which is the oil phase, the organic phase, you'll have your polymer, which has the drug dissolved in it. So when you mix these two and give it a lot of energy, what will happen is, your polymer is going to trap your drug inside this, because everywhere else water is in a very high quantity, so there will be small oil droplets which have the drug inside. Oil droplets meaning the polymer droplets, which is hydrophobic in nature, and also your drug is a little bit more hydrophobic. So your drug would be trapped inside the polymer, and everywhere else surrounding these particles we have a lot of water. So these particles, being hydrophobic, they will try sticking to each other. They don't want to interact with water. That is what hydrophobic means, right? They don't like interacting with water. So in an attempt to avoid water, they'll start aggregating to each other. So we don't want this aggregation to happen, because then the whole purpose of having micro and nanoparticles is defeated, right? You want microparticles, distinct microparticles, wherein you will have some amount of drug encapsulated in a polymer layer. So to avoid this aggregation, we use surfactant. So what will surfactant do? Surfactant will form a very thin layer over surfactant will attach to the surface of these particles, and they will start repelling each other. So as a result, very less aggregation will take place. So your microparticles will be very distinct in this particular oil in water emulsion. So this is what a single emulsion process is. And once you start evaporating this solvent, then so after evaporation of this solvent, you will get dried particles. So your dried particles will be nothing but a polymer encapsulating the particular drug. So to avoid, so you use surfactants. For example, PVA. PVA is nothing but polyvinyl alcohol, which is a very widely used surfactant. You use this in this water phase to avoid the aggregation of these particles. So why do first of all particles aggregate? Because they have a lot of surface tension on their surface. So to reduce that, if you reduce the surface tension, there will be very less aggregation. So the correct answer to this question is, what is the use of surfactants in single emulsion method? It is to reduce the surface tension. Is this clear? If there are any doubts with this question or with the explanation, please let me know and we can go over this again. If not, I'll move on towards the next question. Okay, looks like No doubts. Let's go to the next question then. Next is, what are the disadvantages of using emulsion based methods for synthesizing nanoparticles? We have to choose two correct options. If you people have read through or have studied the emulsion based methods in detail, you'll be able to answer this. If anybody has the answer, do let me know. If not, then we'll of course discuss. Anybody wants to volunteer for this question? No responses? Okay, somebody is answering. Two people have answered first and last. That is high polydispersity and difficult to synthesize particles below 150 nanometer. Okay, thank you both of you for answering. Anybody else wants to give it a try? No one? Okay, let's look at the answers. So both of you are correct. The correct answer for disadvantages of emulsion based methods is, first of all, it gives a very high polydispersity. As I was saying, high polydispersity meaning there's a lot of variation in the size of these particles that are being synthesized by emulsion based methods, and also it is difficult to synthesize particles below 150 nanometer. So if you have come across this slide, these are the shortcomings of emulsion based methods. High polydispersity, there is a chance of biomolecule denaturation, because your whatever drug you have, if it's a protein, it will get exposed to a lot of polymers, and since the polymer is dissolved in the solvent phase, it will get exposed to the solvent as well. Also, a very high amount of energy is required to break these emulsions into tiny droplets, tiny sizes, right? So there is a chance that your biomolecule will get denatured. Also, these methods, though they sound very attractive, they have very low encapsulation efficiency, especially for water soluble drugs, because your emulsion based methods, the single emulsion based method, as we saw, it's an oil in water droplet, right? So your drug has to be a bit more hydrophobic to be dissolved into the solvent and the polymer mixture. So if you have a water soluble drug, initially there will be very less solubility. Since it has less solubility, very less amount of drug would get into this polymer membrane. So there are chances that your particles will have very low encapsulation efficiency. And also it is difficult to synthesize sizes below 150 nanometer, because no matter so at some point your energy is going to be getting saturated. So no matter how much amount of energy you give, there'll be a point wherein you can't go below 150 nanometer. The particles will be either 150 nanometer or above that. So these are few shortcomings of the emulsion based methods. So based on depending upon the options here, we have two correct answers for this question: high polydispersity, and it is difficult to synthesize particles below 150 nanometer with emulsion based methods for synthesizing nanoparticles. Is this clear? Are there any doubts in this, or if anything is unclear, we can discuss again. If not, then I'll move on towards the next question. Okay, let's look at the fifth question. So consider that we are encapsulating DNA plasmid along with a hydrophobic drug, for example rapamycin in PLGA microspheres. Choose the best choice of synthesis that would have higher bioavailability for both of these moieties. Is it so we need higher bioavailability both for DNA plasmid as well as rapamycin, which is already in a PLGA microsphere. So, which is the best method that we can achieve this? Is it single emulsion with DNA and rapamycin encapsulated together with PEG on the outside? Is it single emulsion with rapamycin in matrix and DNA added after formation that diffuses in the microsphere? Or is it a double emulsion method? So, take your time in reading the first of all the question properly and the options and let me know. If anybody knows the answer, please let me know. Anyone would like to volunteer for this? Somebody is answering. Two people are answering option 4. Double emulsion with DNA in inner aqueous phase and rapamycin in the PLJ matrix. Okay, thank you for answering. Anybody else has any other opinion? Okay, let's look at the answer. Yeah, both of you are correct. The correct answer is double emulsion with DNA in inner aqueous phase and rapamycin in the PLG matrix. So this is how the double emulsion particles synthesized through double emulsion look like, right? So they have this polymer uh, membrane here, inside which you will have your first of all water soluble drug, and if you have a hydrophobic drug that can go in this polymer membrane, and around it you have this large aqueous layer, uh, aqueous environment, which will have your surfactant to avoid aggregation of these particles. So what is the question saying? We have to encapsulate first of all DNA plasmid along with a very hydrophobic drug, that is rapamycin. So definitely in a double emulsion method you can, first of all in single emulsion method you won't be able to put both of these moieties together, because single emulsion we just saw right now, right, that the drug has to have some solubility in the polymer and the solvent phase, so it has to be a little bit more hydrophobic. So in that case only rapamycin would we would be able to encapsulate only rapamycin through a single emulsion method. So both of the first options, first and the second option, become invalid. Now the look at the third option. The third is double emulsion with rapamycin in the inner water phase and DNA arranged on the surface of PLJ. So what would happen in this case? First of all, rapamycin is a hydrophobic drug, so it will be very difficult to get it into the inner aqueous phase. You will have very less loading efficiency. And on the other hand, if you have DNA arranged on the surface of PLJ, since DNA is very sensitive molecule, if it gets exposed outside this particular particle, then there are chances that your DNA will be getting denatured. So to avoid this, the only right option is option 4. You will have to encapsulate DNA in the inner aqueous phase, and rapamycin can go in the PLG matrix, which is hydrophobic in nature. So in that way you will have higher bioavailability for both DNA as well as your rapamycin. Is this making sense? Is this clear? If it's unclear, I can explain it again, or if there are any other doubts regarding this method, we can discuss that as well. If not, then I'll move on towards the next question. Okay, I think there are no doubts with this. Okay, sure. Then let's move on towards the next question. Next is select the correct option, rather two correct options. So, our nanoparticles with hydrodynamic diameter 6 to 100 nanometer can transverse the endothelium. Proton sponge effect is used to help particles escape degradation from lysosomes. Endosomal compartments have pH in the basic range, and microparticle formulation has the potential to improve the shelf life of drugs. So, which of these two of these options are correct? You people can let me know if you know the answer. If not, then we'll discuss. Anybody would like to volunteer? Anybody wants to answer this? Okay, 2 and 4. Okay, let's look at the correct answer. Yeah, you're correct. The correct option is proton sponge effect is used to help particles escape degradation from lysosomes, and microparticle formulation has the potential to improve the shelf life of drugs. There is some mistake with this question because option 1 is also correct. Nanoparticles which have hydrodynamic diameter between 6 to 100 nanometer, they can cross the endothelium. So just with this question, there's some printing mistake here. First option is also correct. Also, proton sponge effect is used to help particles escape degradation from lysosomes. So, what is proton sponge effect? So, what happens is when you deliver these micro or nanoparticles, they are taken up by the cells through a process called as endocytosis. And once they get inside the cell, they end up in compartments such as endosomes and lysosomes, where the environment is very harsh. Because it's a foreign particle, your cells will think that this is something foreign and I need to eliminate this, I need to destroy this particle. So in the endosome and lysosome, the pH is very low. It's a very acidic environment, and in that case, your drug will get denatured. First of all, your drug will stop working and the particle will completely be chopped up. So to avoid that, what we can do is we can try designing the particles in such a way that they carry secondary or tertiary amines. What this means is they'll have functional groups such as NH and NH2. What they do is they try sequestering most of the protons that would be there in the endosome or lysosome. As I said, it's acidic in nature, so there are a lot of H plus ions roaming around in these compartments. So your secondary and tertiary amines will try sequestering these H plus ions. Now what will happen because of this? The pH will get disturbed, the pH of endosome will get disturbed, and the cell will think that something is going wrong with the endosome, so it will try maintaining the pH. In that process, it will try pumping more H plus inside the endosome. So you have your endosome, you have your cell, and outside the endosome there is the cytoplasm, right? So the cell will try putting more H plus from the cytoplasm into this endosome. Now there will be a lot of H plus ions in the endosome. To balance that, a lot of chloride ions, Cl minus ions, would be pumped inside the endosome. Now what will happen in this process? Now your endosomes is having a lot of H plus ions, it is also having a lot of Cl minus ions. So there is a lot of ionic strength that gets created inside these endosomes. So outside endosomes very less ions, inside endosomes lot of ions. So water will start leaking from the cytoplasm into the endosome. In that process, so the membrane of the endosome has some capacity to hold in water. If more and more amount of water starts going in, it is going to burst your endosome, and as a result the particles will very quickly come out of the endosomes into the cytoplasm. So this is one of the techniques that is used to help these particles escape from the endosome. So that is the proton sponge effect. It is used to help particles escape degradation from lysosomes. And the third option, endosomal compartments have pH in the basic range. We just discussed this, right? So they don't have pH in the basic range. The pH is in the acidic range, highly acidic. Last option is microparticle formulation has the potential to improve the shelf life of drugs, which is also true. So for this particular question, both all the three first, second and fourth options are correct. Is this clear? Okay, if there are no doubts, I'll move on towards the next question. Okay, the next question is liposomes are made of phospholipid bilayers and membrane fluidity is important for drugs to pass through them. Select the correct option about membrane fluidity. We have to select one correct option. So, does the fluidity depend on the cholesterol content of the bilateral membrane, or as the temperature increases, will fluidity increase, or is it independent of the length of fatty acid tail, or as the temperature increases, fluidity will decrease? So, one correct option. If anybody knows the answer, please let me know. Any volunteers? Two people are answering as option 2. As the temperature increases, fluidity will increase. Yeah, you both of you are correct. That's the correct answer. So as we were talking about liposomes, right? So liposomes are nothing but they're synthetically made phospholipid bilayer, inside which it has an aqueous core wherein you can dissolve your you can encapsulate your water soluble drugs. So this membrane that is made, so it is fluidic in nature, and as your temperature increases, the fluidity increases. So this is one way where this is one phenomenon how the drugs get released from this particular liposomes. So fluidity meaning these bilayer, these bilipids, they are not very strong and very tight in their formation, so there is some amount of fluidity here, and that's how the drug will get released. So if you look at the options, the fluidity definitely depends upon the cholesterol content. That is what also happens in our the bilayer membrane that our cells also have. And as the temperature increases, fluidity will increase, and it indeed depends on the length of fatty acid tail. So this option 3 is incorrect, option 1 is incorrect, and as we already saw that as temperature increases, fluidity will increase instead of decreasing. So if you consider the options, only option 2 is the correct one to define these phospholipid bilayers. Is this clear? No doubts here? Okay, then I'll take up the next question. Next is, which one of these is not an advantage of nano or microparticles? So we have to essentially tell them what is the disadvantage of having nano and microparticles. So uh, read through the options and then we can discuss. Any volunteers for this question? Okay, people are answering option 2. It elicits an inflammatory reaction in the host. Okay, thank you for answering. Any other response? No? Okay, let's look at the answer. Whoever is answering in the chat box, you're absolutely correct. The correct answer is it can elicit inflammatory reaction in host. That is one of the disadvantages of having nano and microparticles. So let's look at the options. It's injectable. Definitely it's an advantage. We don't have to do a surgery to implant these particles into the patient's body. So being micro and nanoparticles being injectable is actually an advantage. Possible way of delivering large hydrophilic molecules inside cells is also an advantage, because as we were discussing right now, the cells have a phospholipid bilayer which is essentially hydrophobic in nature, so it's difficult to deliver hydrophilic molecules inside the cell. So in that case you can make use of these nano and microparticles. It's again an advantage. Particles can be adapted for targeted delivery to cells and tissues. Yes, you can do that. And it also improves the stability of the drug in vivo. So the only disadvantage of having micro and nanoparticles is it can elicit inflammatory reactions in the host. So some of these points are also listed in this slide. You can go over it later on if there is a confusion. Is this question clear? And also the explanation? If there are no doubts, I'll move on towards the next question. Thank you for people who are answering in the chat box. I may not be able to look at the answers. Okay, then let's look at the next question. Which one of the following parameters does not affect microparticle synthesis using double emulsion method? So read through the options, and if you know the answer, please let me know. Anybody? Okay, 3 people have answered last option, which is none of the above. All of you are correct. That's the correct answer. So all of these factors actually, all of these parameters actually affect the synthesis process when we are dealing with double emulsion method. So you can read the detail of this method here. So in double emulsion method, it definitely so all of these parameters will start affecting your particles. First of all, it's the polymer type and the molecular weight. That is the main constituent of your particles, right? So you have this polymer membrane, and it's like it forms a shell, and inside at the core there is your drug. So what type of polymer you are using and what is the molecular weight of these polymers will affect your particles. Also polymer constant, how much amount of polymer you're putting in the oil phase, the type of drug and the method of incorporation will affect your particles. Whether your drug is solid, whether it is liquid, whether it's hydrophilic or hydrophobic, that will determine where your particle is going to land up in the inner phase, or will it be in the polymer membrane. Your organic solvent that you're using, and because the polymer solution is made in this organic solvent, right? So you need to know what is the solubility of your polymer in this particular organic solvent. Then the type and amount of surfactant in the external phase. So this helps in avoiding aggregation. So you need to know how much amount of surfactant to put. The internal aqueous phase to organic solvent ratio definitely matters. Rate of stirring and type of mechanical force that you are using to break these particles into tiny particles. So if you want a very lower size of particles, you have to give more amount of energy, that is increased speed of stirring. The evaporation rate, at the rate at which the solvent is going to be evaporated, the temperature, pressure, and also the volume in which all of these reactions are being carried out. So all of these parameters will affect the type of particles that you are getting. So the question was, which of these does not affect microparticle synthesis? The correct answer is none of the above. Is this clear? Because all of these parameters will start affecting your particle quality. Is this making sense? I think that was the last question I had for you for this assignment. So if anybody wants me to repeat any of the previous questions, I can do that. If not, then, or if there are other doubts or any other concepts that you'd like me to explain again, we can do that. If not, then I can stop the meeting here. That's all I had for you for today's session, and we can meet next Tuesday at 6 o'clock. Does anybody have doubts? If not, then I'll stop the meeting here. You can also let me know if there are doubts in the next session as well. You can go through the material of this session, which I'll be uploading. And if there are doubts, then we can discuss in the next session as well. If not, then I'll stop the meeting here. Is that okay? People who are still in the meeting, do you have any doubts to discuss? If not, you can log off. Thank you for joining today's session. Is that all? Shall I stop the meeting here? Thank you. Thank you for joining. Okay, see you people next week, next Tuesday. Thank you.